Hey guys, welcome back to another video of my channel. Today we are going to discuss another very important topic from epistasis. What is epistasis? Basically, it is the non-allelic gene interaction. Okay, now let's understand what the topic says. Non-allelic gene interaction, right? Earlier, Mendel gave the concept that oh sorry, one gene, according to Mendel, control only one trait, right? So when we got into dihybrid cross, it gave a ratio like 9:3:3:1, which showed independent assortment. But later on, when Batesian and Punnett they got into research, they found that two different genes can sometimes code for the same phenotype, right? How we will understand this? But two different genes can sometimes show cumulative effect like this action of X and action of Y can all result into C. This was considered as epistasis, that is non-allelic gene interaction, right? Two different genes code for a same phenotype, that is a color or a shape, like we'll consider them. And now let's move how. Batesian and Punnett showed that two non-allelic genes code for the same phenotype. I told you. Now let's check how they code for the same phenotype, right? There is a colorless white precursor. C gene acts over this, forms a intermediate precursor. Here it is a white flower only. After the action of intermediate white precursor, it shows white flower only. But when the P gene acts over the intermediate precursor, it forms purple color. Sometimes, if the intermediate precursor doesn't act, right? If the P gene directly acts on the colorless white precursor, though it might form some different color, right? Now let's see how. See here. See, this is the dominant, this is the dominant, this is the recessive, right? Here we will see that if if the C gene is if the C gene acts upon clearly, then it forms the intermediate precursor, and after the action of C gene over P gene, it shows purple color offspring, right? So the action of dominance of both C and P can only form purple color, right? Where the C or P is not dominant, it will show white color only. So we have seen that. I'll tell you this thing. Wait, we'll consider it here that C, capital P. We have seen here capital C shows 9 progenies which carry one capital C and one capital P. Here we can have small c, capital C, or small p, capital P, right? But it will show purple color only because C is dominant over small p and P is dominant over small p. Rest all the progenies will form white trait, right? Because they have one or both the genes in the recessive form. Now let's see the cross. Small c, capital P is crossed with capital C, small p. It forms all these progenies on selfing. It can form these. Both of them can independently form four of them. So we held a cross, and all those containing at least one capital C, capital P showed purple color flower, and rest all showed white flower. Right? Here we have taken this. I know you might be thinking that the topic is getting a bit difficult. We'll move further, you'll get to understand. Okay. You can't get the topic in once because it is some very difficult topic, one of the difficult topic and very important topic for the CSIR point of view. Okay, so in epistasis we consider four different cases, that is dominant epistasis, recessive, duplicate dominant and duplicate recessive. See what is seen in dominant epistasis. In dominant epistasis, dominant form of one allele masks the dominant form or the recessive form of the second allele, right? So, if we have, let's assume there are two genes A and B. If we have a white precursor, if gene A acts upon the white precursor, it forms purple progeny. But if gene B acts on it, it forms the red progeny. So, gene B may be present or not. A may be present or not, right? So, it forms this two different progenies. And where both the genes are in recessive form, it will form the white precursor only. Now let's see. Here we have seen A gene has got dominance, B has got dominance, right? So earlier when gene A acted, we got purple flower. So G here we'll get purple flower. B could not show its dominance because the A gene has already shown the dominance. Here we have seen purple flower because A is dominant. Here B is dominant, so we'll get red flowers. Here none of them is dominant, so we get white. You might be knowing na, how we got these ratios. You might be knowing it when we get a dihybrid cross, then we get these ratios. If you have any doubt, then pause the video right here and consider a dihybrid cross like this, somewhat this. Okay, in 9 cases, like these, we have got both the traits as dominant. In 3 of them, we get one dominant, and in 3 of them, we get the other one dominant, and in one, we get only none of them dominant, right? So this, so here we have to see gene B works only when A gene is not present, right? Gene B works only when A is not present. So only red color will be seen when A is completely recessive. See, A here is recessive, so we got red color. Otherwise, here A is dominant, so we didn't get B because A shows dominance over the expression of B, both dominant and recessive. Here A is like, ki, I don't care. B is in dominant or recessive, I don't care. I am here to give my color, so I'll give purple color. So we got the ratio for purple. That is purple 12 flowers, red 3 flowers, and white 1 flower. Okay. So here we got recessive epistasis as our second case, also known as the supplementary epistasis. So here the recessive form of one allele dominates over the dominant or recessive form of other allele. So let's see how. Here we had the white substance as the precursor. Then gene A acts over the white substance and forms the red one. Then if complementary to gene A acts gene B, then it gives purple color. But here the recessive form of small a masks the effect of gene B and absence of gene A. Basically, any case we can consider, we have to see, take one gene like A and its recessive form is epistatic, means it can mask both capital B and small b. If we have considered that A is showing, small a is showing purple flower, then we will not consider that it is with this, or it is with this, or it is with this. In all these cases, we will get only purple flowers, right? So first we'll consider that small a is giving out white flowers. So everywhere where we get small a, we will get white flowers only, right? And if capital A acts over white precursor, then it forms the red flower as the intermediate. And if it both the genes are in dominant form, it forms the purple color. Basically, you have to learn that recessive epistasis means recessive of one form mask the expression of the dominant or recessive or heterozygous form of the other gene. So we get up to this ratio 9:3:4. Purple 9, red 3, white 4. I know the topic might be getting a bit difficult for you, but this is what it is. You'll have to learn the topic or revise it 2-3 times, then you will get to understand. For once, it was difficult for me too. Here we will see duplicate dominant epistasis. What you get by duplicate? Duplicate dominant means it is mimicking dominant. It is not completely dominant, but it is mimicking. If alleles of both the genes produce same phenotype without any cumulative effect. Now let's see how. Duplicate allele of either gene allows the conversion of reaction to produce wild phenotype. Now again, if A acts and B acts together, then they form purple. Missing of any of them, right? If single one also acts, if only A acts, then also it is going to form the purple flower. If only B acts, then also it is going to form purple flower. If both of them acts, they will also form purple flower, which shows that we were thinking that one gene is dominant over other, but no, it is duplicate dominant. So both the genes are dominant, whether present individually or together. सो सिंपल फैक्ट इज कि अगर ए प्रेजेंट होगा डोमिनेंट फॉर्म में तो भी पर्पल कलर आएगा, बी प्रेजेंट होगा डोमिनेंट फॉर्म में तो भी पर्पल कलर आएगा, और अगर दोनों डोमिनेंट फॉर्म में प्रेजेंट होंगे तो भी पर्पल कलर आएगा. सो ओनली we have left with one case where AABB is recessive, then in that case only we get white flower, rest all are purple. This is 15:1. Now we get up to duplicate recessive epistasis. Again, duplicate recessive, which shows that small b dominates over capital A, capital B. That is, both of them, when expressed, can mask the expression of capital A and capital B. In recessive, we considered only one gene's recessive form that was masking the expression of other. But here, recessive form of both the genes is masking the expression of the dominant traits. Here you have seen, we have written capital A dash small b, capital B dash small a, capital B dash, capital A dash small b, and small a, small. Here we have got capital A, small b, and both small b. So in all these cases, we will get the same white. Only when both are dominant, then only we have got different case. That is 9:7. 9 purple flower, rest all. Where one or both the genes are in recessive form, we get the white flowers. That is the default case. Only the action of both the dominant flowers shows purple color. Now here is a summary shot of that we have got this dihybrid ratio, out of which first we have the dominant epistasis, then we have the recessive epistasis, then we have the duplicate dominant, then we have the duplicate recessive, right? These are the ratios we got, and this is allele of one gene dominates, small a dominates over the capital B and small b, A and B. Capital A and capital B are duplicate dominant, that is, they are dominant over small b both. Small a and small b show duplicate recessive. That is, also remember that this shows the supplementary epistasis and this shows the complementary epistasis. Right? This is a summary of whatever we have learned till now. Right? We have to understand that what it shows. Dominant shows the dominance of one of the gene over the other. Recessive shows that recessive form of one gene masks the expression of other. Duplicate dominant shows that both dominance of one or both the genes will show the effect over the other, and duplicate recessive shows that the recessive form of one or the other shows the effect together. Now I have got a question. Pause the video, l- understand the question, and then wait. I hope that you might be able to see the complete question. If not, wait a minute. This is the question, and these are the options. These are the options. Right? Now pause the video and get into the question. Now let's discuss. I hope you might have got the answer. Two interacting genes independently assorting were involved in the same pair. Absence of either of the gene function leads to the absence of end product. A dihybrid cross involving two genes is carried out. What fraction of F2 will show presence of end product? Now it says that we have got this, and this. Absence of any of them. If A gene acts, then we get Y, and if B acts, then we get Z. So in absence of A or B, we don't get Z. So this is the form, right? So this is clearly an example of dominant epistasis. That dominant form of both the genes involved shows helps in the formation of the end product. So this is asking that what fraction of the F2 will show the presence of end product. We have seen, oh sorry, that we got capital A dash capital B ratio small a capital B dash ratio capital A dash small b ratio small b small b, right? 9 ratio 3 ratio 3 ratio 1. So here if both A and B are dominant, then only we will get Z. So here both A and B are dominant, so we'll get this, as this will show Z. Here only B is dominant, so we will not get Z. Here only A is dominant, so we will get only as product. Here none of them is dominant, so we'll get X only. Here also we will get X only because A is not dominant. So to get what fraction do we get Z? This is 9. 9 over 16. So I hope it might be clear. So we got the answer 9 ratio 16. Now this is the other question. Look at the question, understand it, and tell me the answer in the comment section. Pause the video, check, understand the question, right? This is your question, and this is the option you have given. I hope you might have got the question. Now let answer. Now let's check. Following is a hypothetical pathway responsible for pigmentation of leaves. The pathway is controlled by two independently assorting gene A and B. Mutant alleles A and B code for a non-functional protein. Non-functional means the default stage, right? Obviously, small b code for the default stage or the non-functional stage, or we can say majorly in cases white, right? So here we have given this statement also. X is a white substrate, Y is the yellow intermediate, Z is the green product. Here A acts forms the yellow intermediate, B acts from the green product. So again we'll write the same ratios, right? You can speak along with me. Capital A dash small b ratio small a capital B dash small b 9 ratio 3 ratio 3 3:1. So, okay, we will check where all do we get capital A, right? I- Wherever we have got capital A, we will get the yellow intermediate. Here, yellow intermediate, these will be the yellow ones. Yellow, okay? Where only B acts. The action of B is only expressed when A has shown as effect. So dominance of B is of no use until and unless A is capital. So it will show the default stage or the non-functional state white, right? Here also we'll get white because both are in recessive. Here we will get green product because dominance of both can be seen. So here we'll check in the options which is, that is 9 of green, 3 of yellow and 4 of white. Which option you think is most expected? Tell me. I think the first one is right. I hope it might be clear. If you want me to drop more question videos like this, please drop down in the comment section. I hope you liked the video. Please like, share and subscribe. Thank you.